Good afternoon, distinguished guests, ladies and gentlemen. Welcome to join us at the Laureate Lecture of 2024 Tang Prize in Biopharmaceutical Science. My name is Angela Liu, your MC for today's event. To begin with, we would like to invite to the stage the host of this lecture, Professor Wang Huijun, academician of Academia Sinica, to give us the introduction. Ladies and gentlemen, please welcome Professor Wang.
Good afternoon. My name is Andrew Wang. I'm the host of this lecture here. We have three great scientists to give lectures in this biopharmaceutical science in Tang Prize laureates. And first of all, I'd like to mention that today is a very special day. It's Teachers' Day in Taiwan. So we are all teachers. So the students usually give us a lot of blessings and so on. But sometimes they give us some gifts, so that's nice. But anyway, today, I think we invite three laureates to give us their contribution in this big prize here. Is on the discovery of GLP-1 7 to 37 as an insulinotropic factor and the development of GLP-1 7 to 37. That's a very unusual number, 7 to 37, based anti-diabetics and anti-obesity drugs. As many or some of you know that this GLP-1 industry is blooming, and the company who developed this, Novo Nordisk, in Denmark, right? Yeah. So the its market price, I was just told by Chi Hui-wong, our former president, that it's even bigger than Taiwan's GDP. So one drug can make a lot of money. But today, I think the three laureates are Joe Habener. Joe was my actually friend in the early 70s when he was a junior physician at MGH, and he came to MIT to do some basic research, so we sort of overlap a little bit. And Joe is now retired from MGH, but he made his very early discovery on GLP related biology, and that led us to this very important discovery in this GLP-1 7 to 37 drugs. And during the early 70s, Svetlana Mojsov, she was the director of a core facility at MGH and the Howard Hughes Medical Institute there. So she worked on the peptide synthesis, and as you will hear from her talk, that she described how she discovered the 7 to 37 is very important. And last but not least is Jens Holst from Denmark, and he independently worked on the GLP-1 from a real biological species and discovered the similar things, that it can induce some insulin effects. So without further ado, I think we'll have three lectures consecutively. The first two, I believe, is in video because they could not come to this event. And then finally, Dr. Holst will give a lecture in person. And after that, we'll have a very short summary. Thank you very much.
Thank you very much to Professor Wang for the introduction. Thank you. Please take your seat. And now, ladies and gentlemen, let's give our full attention to Professor Habener.
Good morning, everybody. I'm pleased to be here and this lovely day and to talk about my favorite subject, glucagon-like peptide 1. So we can just start with the first slide. The next slide is a a preview of what I'm going to talk about for the next 30 minutes or so. And that's the discovery and the utilization now of GLP-1, the innovation moment. It's GLP-1 is a 30 amino acid peptide incretin hormone made in the intestines and secreted with meals. It stimulates glucose-dependent insulin secretion, lowers blood glucose in type 2 diabetes, and delays gastric emptying, and thereby promotes weight loss and obesity. And a third use for GLP-1, it appears now, is a treatment for cardiovascular disease risk. It lowers disease risk in people with cardiovascular disease. Next slide. You can go to the next. The topics I'm going to cover are three, and mainly the discovery phase of the research. Again, I've done at the Mass General Hospital, and the two other topics are some comments about the impact on health and some comments about the possible future developments in GLP-1 physiology and medicine. Next slide. First, the discovery. Next slide. The goal of our studies was a curiosity to know whether there are prohormones, that is, progenitors for the pancreatic hormones glucagon and somatostatin. The rationale for this embarking on these studies is that it was known that there are precursor proteins encoding the hormones insulin and parathyroid hormone, PTH, and opiomelanocortin, which is a prohormone that encodes more than one hormone. It encodes melatonin and ACTH. And the second rationale was already there was evidence that there are large forms of intestinal glucagon, namely a hormone called glycemtin and one called oxyntomodulin, which glycemtin has an extension of a peptide on the N-terminus of the glucagon, the left, and oxyntomodulin has a peptide extension on the right, suggesting, indicating that there's very likely a larger molecule in which the glucagon is encoded. Next slide. The approach we used was recombinant DNA cloning, and that involves isolation of the islets from the rat pancreas. And from that islets, we prepare RNA, and then the RNA is used to make cDNA using the reverse transcriptase enzyme that had been recently discovered by David Baltimore. And then the cDNA is then cloned into bacteria and can be decoded by the genetic code to give the amino acid sequence, the protein. And it has the great advantages of avoiding sequencing of the proteins, which is very laborious and time-consuming and expensive. And it expedites obtaining the complete sequence of very large proteins via the decoding of the cDNAs. The next slide. So the first step is to isolate the islets from the pancreas. And this is a diagram showing the anatomy of the mammalian, including humans, the pancreas, which consists really of two organs, an exocrine pancreas and an endocrine pancreas. And the endocrine cells are scattered about in 1,000 little small spheres, which contain the endocrine cells that make hormones, whereas what's shown in pink here is the islets. They call them islets of Langerhans, which contain the endocrine cells, which are three major types: the beta cells, shown in green, and the alpha cells in blue, and the somatostatin cells in red. And as I mentioned earlier, they've been shown already from work from Don Steiner's lab in Chicago that insulin is made as a pro-insulin, a precursor, a pro-hormone. So we asked the question again, whether the somatostatin and glucagon hormones were made as precursors. So, so our goals were to isolate these islets and extract them for the messenger RNA to make the cDNA, to clone the coding sequence for the genes, the messenger RNA for the glucagon and somatostatin. And so the next slide shows that there was a change of plan in our experimental approach, that the NIH had declared a moratorium on recombinant DNA research, even warm-blooded animals, for example, the rat. So that plan was not possible to carry out. The moratorium was declared in Cambridge and was intended to be extended to Boston. So the research committee did not approve our protocol to start the research. So the next slide, we went, it was an indication to go on a fishing expedition. And our goal was to capture the anglerfish, the so-called Atlantic anglerfish, because it's a cold-blooded animal and would be allowable to carry on the research. So the next slide shows the great advantage of the anglerfish, thanks to the moratorium, is that the endocrine cells are not scattered in islets that you have to isolate from the exocrine pancreas, but they're in a separate organ called the Brockman body. And it makes it easier to and more efficient to extract the RNA from an organ that is enriched, so highly enriched in endocrine cells. So the next slide shows that our initial testing of the quality of the messenger RNA that we extracted from the anglerfish. We used a cell-free translation system that had been developed at MIT by Brian and used the answer to glucagon and somatostatin to immunoprecipitate the protein products of the cell-free translation, and found that it's very promising that the precursors, the proteins and translated by the messenger RNA were, for both glucagon and somatostatin, were much larger than the hormones they encoded. For 14,000 molecular weights, 14.5 and 12.5 kilodaltons, and for the glucagon. And yeah, next slide. So the eureka moment was when we got the sequencing and found that there were indeed the glucagon was a small peptide encoded in the in the middle of the so-called pro-glucagon, and the sequence of glycine was clearly displayed there. And what was unique was there was a second hormone-encoding peptide, which we called glucagon-related peptide. It was structurally within the glucagon superfamily of hormone peptides. And and these various peptides are cleaved at two basic residues as shown by the arrows, either arginine or lysine, and arginine-lysine pairs. And suggesting that this is a prohormone that's giving rise to more than one peptide. It's at least two called the glucagon and the glucagon-related peptide. And the next slide shows that the sequences of the GRPs, there was actually two GRPs, non-allelic genes, two genes for the proglucagon, and we call them GRP1 and GRP2, and they're very similar, but the similarities in the hormone family that's encoded in that make up the glucagon superfamily of peptide hormones. That most closely resemble the hormone called gastric inhibitory peptide, which is shown at the bottom, and it has the sequence of 60% or so similar to gastric inhibitory peptide, which is already of interest because the next slide shows that the GIP was shown previously by Werner Creutzfeldt in Germany to be an incretin. Then the incretin concept suggested that it showed that there's a factor secreted from the intestine that augments glucose-stimulated insulin secretion, and that the incretin effect was shown by Creutzfeldt to be and predecessors that an oral glucose load is a more potent stimulus of insulin secretion than is an equivalent glucose load given systemically by intravenous. So, and that it was further shown by immunoneutralization experiments in the Creutzfeldt lab that the gastric inhibitory peptide was an incretin. It augmented glucose-stimulated insulin secretion, but only partially. It didn't account for the full incretin effect. The next slide just shows you what the incretin effect is. And to the top panel is the glucose given either orally or systemically, so that you give an oral glucose to a patient or subject volunteer and measure the glucose level, and then mimic the same identical glucose level in the blood by an intravenous infusion, and then measure the insulin response to the glucose infusion, and one see that the oral glucose is much more efficient at stimulating insulin secretion than is the systemically intravenous dose of glucose. So that's called then the incretin effect. And it's now been shown in humans the effect of GIP and GLP receptor agonists, that glucose is 25% of the stimulus for insulin secretion, and gastric inhibitory peptide and GLP-1 are 45 and 30% respectively. And so the total incretin contribution to the incretin effect of GIP and GLP-1 is 60% and 40% respectively. Next slide. Subsequent to the identification of the glucagon-related peptide in the fish system, we went on, as did many others in the field, and including determining the sequence of the proglucagons or pre-proglucagons that for mammalian species, and we worked on the rat protein and isolated the DNA encoding both the gene and the messenger RNA, the cDNA, for rat pre-proglucagon. And interestingly, the hormonal and functional components of the protein, the proglucagon, are encoded in separate exons, very nicely segregated, with the GRPP, the glucagon-related polypeptide, glucagon, GLP-1, and then a second GLP called GLP-2, which turns out to be a growth factor for small intestinal stimulus for small intestinal growth. The next slide. It shows the interesting example of how the cleavage of the proglucagon involves an alternative post-translational processing by specific different prohormone converting enzymes, prohormone converting enzymes, called PC2 and PC1/3. And the PC2, for example, liberates glucagon, leaves the GLP-1 and GLP-2 peptides intact as a major proglucagon fragment in the alpha cells of the pancreas, whereas in contrast, in the intestine, the separate prohormone convertase is expressed and envelops and cleaves the proglucagon into liberating the GLP-1, just shown in the red, and leaves the N-terminal sequence, the glucagon-related polypeptide and glucagon intact as the glycentin moiety. And so the two hormones have different functions. The GLP-1 is an insulinotropic function that's involved in feeding behavior, and the glucagon is an anti-insulin hormone involved in fasting. It stimulates glucose production by the liver, raises blood glucose, whereas the insulin made in the pancreas stimulates disposal of glucose and lowers blood sugar. So next slide shows an interesting conundrum that we encountered after identifying the GLP-1 peptides, that there's a possibility to show that there are two possible sites where GLP-1 might begin in its translation of the messenger RNA, because there are two basic residues, lysine and arginine, K and R, and it's cleaved at between the arginine and histidine, which should be the much preferred cleavage site based on the understanding that double basic residues were involved in sites of cleavage by the prohormone convertases. But then there were 6 amino acids later. There was another single arginine, and again a histidine, that would begin the translation of the glucagon-related, the glucagon-like peptide. But it's unexpectedly, as I'll show you in our studies, they did collaborated with Dr. Mojsov, Svetlana Mojsov, and we found that the active peptides are the peptides that are cleaved at the single arginine, and so at 7 to 36 and 7 to 37 are the active peptides. And I should mention that the C-terminus comes in 2 versions. One is an arginine amide, and the other is a glycine extended. So it's a simple amidated and a glycine extension, but they both have the identical activities. But the next slide, there are two studies that were done that really determined which was the active peptide, and one was using insulinoma cells in vitro that are a good model for endocrine pancreas cells, and so we could compare the effects of 1 to 36 and 7 to 36, the 2 GLP-1 analogs, the N-terminal extended 1 to 37 and 36, or the N-terminal truncated, the shortened to 7 to 36 or 37. And in this initial studies, the cyclic AMP formation could show the studies that Dan Drucker did in the lab, that the shortened truncated peptides, 7 to 37, stimulated cyclic AMP formation in the insulinoma cells, whereas the 1 to 37 had no effect except extremely high doses, which probably represented an in vitro conversion of some of the peptide due to cathepsins in the serum, in the media. And but importantly, the 7 to 37 peptide both stimulated proinsulin messenger RNA levels and, importantly, insulin secretion. But the second study was done, the next slide shows in vitro and vivo studies, and that is a model that we did in collaboration with Gordon Weir at the Joslin Diabetes Center, and we studied the secretion of insulin from the perfused rat pancreas. So it's sort of an in situ, in vivo model insulin secretory dynamics. And so we see at the lower panel, on the left, the 7 to 37, and on the right, the 1 to 37. And the 7 to 37 stimulates insulin secretion at 50 picomolar, shown on the left curve, which I don't have a pointer, but just show right. The curve goes up and markedly increased at 5 nanomolar. So it's a potent stimulator of insulin secretion, where even at 5 nanomolar, which is 100 times higher concentration than 50 picomolar. The 7 to 37, the 1 to 37 has no effect. And even up to 50 picomolar, there's virtually no effect. Okay, so next slide. And then finally, we did a nice study collaboratively with David Nathan at Mass General, and to study the effect of GLP-1 infusions in human subject volunteers with type 2 diabetes. The study was done during a meal stimulation test, which is shown on the red down here, the meal. It was a 30-minute meal where the subjects were required to complete a small meal in 30 minutes. And during that time, they were given infusions of either GLP-1 or saline. So you can compare a placebo with the hormone at different doses. Interestingly, it became quite evident during the meal test that the subjects who were receiving GLP-1 were getting full faster. They had difficulty finishing their meal. But anyway, one can see that the normal subjects and the type 2 diabetic subjects had quite different responses on glucose, in that the glucose with saline, typically the type 2 diabetes, the glucose rose during the meal. And remarkably, with the GLP-1 infusion, the glucose actually fell slightly during the infusion. And then this corresponds to a robust insulin response with the GLP-1, with the as compared to a very wobbly, meek, minor rise in insulin in the people with the defective insulin secretion, the diabetes, with the saline and placebo. So this is a nice demonstration of the efficacy of GLP-1 in the treatment of type 2 diabetes. Next slide. So go on to the second topic, and that is the comments on the impact of the discovery of glucagon-like peptide 1 on the health of individuals. Next slide is currently there are 3 diseases that are approved for treatment with GLP-1 receptor agonists, and that's diabetes, obesity, and recently approved is cardiovascular disease. Next slide. The mechanisms which was alluded to, how the GLP-1 receptor agonists are effective in treatment of diabetes and obesity and the cardiovascular disease. In diabetes, it stimulates glucose-dependent insulin secretion and reduces postprandial glucose excursion, so that the at the same time, it suppresses glucagon secretion. It works on obesity by delaying gastric emptying, which people get full faster than they can. They don't eat as much. It curbs appetite, probably by a central effect as well as causing feeling of fullness. But the two combined reduces food intake, and as one can understand that by reducing food intake, you reduce energy intake, and it results in weight loss, and it affects on cardiovascular disease risk. It's probably due to effects on reducing inflammation. Next slide. This is important because worldwide prevalence of diseases treated by GLP-1 receptor agonists are pretty compelling need for treatment. The prevalence of diabetes in the world population is about 540 million as of 2020. Yeah, it's about 6% of the population. Obesity is even much greater. About 1.2 billion people are overweight or obese, and cardiovascular disease is about 500 million. So there's large population, groups of subjects in population who are in need of a treatment for these 3 disorders for which the GLP-1 receptor agonists are approved now. GLP-1 agonists that are approved for the treatment of diabetes and obesity and now cardiovascular disease are semaglutide from tirzepatide, or otherwise known as Ozempic or Wegovy, or Mounjaro or Zepbound, which is interesting. Zepbound, tirzepatide is a dual receptor agonist, which actually stimulates the receptors for both glucagon-like peptide 1 and GIP, the 2 major incretins in the body. And so I just wanted to finish with a study, and this is rather remarkable when it was published a year ago. Subjects, 16,000 subjects who were overweight or obese but had no diabetes. So they were simply non-diabetic, overweight or obese individuals, and divided into 2 groups, one of which was treated with semaglutide, and the other group was on placebo treatment. It was a 48-month duration, 4 years, and the endpoints were heart attack, stroke, or death. And remarkably, there was a 20% reduction in risk, which incidentally was seen very early in the study, so that the effects were within the first month or so of the treatment with the semaglutide. Yeah, next we go, next slide. Now there's a few comments about the future where this, the utilization of the GLP-1 receptor agonists might be heading. And so the next slide shows that as time has gone on, as the numbers of patients has increased remarkably during the last 2 to 3 years, particularly, that more clinical trials are being done, and it now has opened the door for meta-analyses of medical records of large numbers of people. It can be divided into groups of, like, say, for example, 10,000 people who are taking GLP-1 receptor agonists, and 10,000 sex and age-matched individuals as controls, and you find out what the incidence and prevalence of the various diseases. And we've already mentioned on the cardiovascular disease risk is decreased by 20%. There's also been evidence now that the other disorders such as non-alcoholic fatty liver disease, NASH, or polycystic ovary disease, or colorectal cancer, and Parkinson's disease, and possibly dementias, and renal failure, and interestingly, addictions. And I think that evidence is coming. It's gathering that that GLP-1 receptor agonists have an anti-inflammatory action, which is so next slide. Yeah, and so the future, much needs to the studies will continue to understand how these different effects are mediated. Just to say, I'm deeply indebted to the multiple people. I think something like 140 postdocs, visiting professors, students, and have participated in the work over the numbers of years that we've been involved in studying on aspects of GLP-1, physiologic, biologic actions, and genetic gene expression and control. And particularly with the early discovery phase of the existence of GLP-1, I think my thanks to Kay Lund, didn't show there, Richard Goodman, Gerhard Heinrich, Dan Drucker, all involved in the early studies, and then subsequent postdocs, people, and my collaborators, Gordon Weir and David Nathan, Darish Elahi has done a lot of collaborative clinical studies with me. And special thanks to John Potts, and for support, I'm particularly indebted to the Howard Hughes Medical Institute, who supported my work for 31 years, and the participation of the National Institutes of Health. Thank you, and thank you very much for your attention.
Thank you very much to Professor Habener. Up next, here is the lecture from Professor Mojsov. Let's give him our full attention.
Very honored to receive the Tang Foundation Prize for my contribution to the discovery of GLP-1, and to have this opportunity to show you my studies which led to that discovery. Now, my interest in the role of peptides in regulating glucose metabolism goes back to the 1970s when I was a graduate student with Professor Bruce Merrivale at the Rockefeller University. We were interested in understanding the role of glucagon in glucose metabolism and diabetes. Parallel experiments in the 1970s were focused on the discovery of incretins. So what are the incretins? Incretins are peptides which were secreted in the intestine in response to a meal, and which then stimulate insulin release from the pancreatic beta cell and thereby influence glucose metabolism. We were interested to understand the biology of glucagon. And glucagon is a 29 amino acid peptide which is secreted by the pancreatic alpha cell and then stimulates the increase in blood glucose level through its action on the liver. Very often, it is referred to as a counter-regulatory hormone to insulin. Now, our approach to understand glucagon biology was to obtain by chemical synthesis large amounts of analogs and inhibitors. And the solid phase peptide synthesis developed by my PhD advisor, Professor Bruce Merrifield, was the method of choice to do that. My PhD thesis was to develop new synthetic strategies to obtain synthetic glucagon in large amounts. And in the next slide, I will show you the principles of the solid phase method and how I applied it to the synthesis of glucagon. So the method consists of attaching the last amino acid in the sequence, in this case a threonine, to a solid support of a resin, and then adding each subsequent amino acid one at a time until the entire sequence is established until the entire sequence is completed. And then by a number of chemical reactions, the protected peptide chain is removed from the resin, and the synthetic material, in this case glucagon, is purified by chromatography. I also went through a second synthetic strategy to develop even a more efficient synthesis of glucagon. And by that time, I've completed these studies. I have known the glucagon sequence backwards and forwards. And this knowledge of glucagon sequence and its biology were critical in my discovery of the active sequence of GLP-1, as I will show you later. Other researchers use molecular biology approaches to study glucagon biology. And in 1983, Graham Bell published the sequence of the human glucagon gene. He showed that glucagon is made as a part of a larger molecule which contained the sequences of two peptides unknown at the time, which Bell named glucagon-like peptide 1 and glucagon-like peptide 2, or GLP-1 and GLP-2. Bell predicted that GLP-1 would be a peptide of 37 amino acids. Other investigators in the field came to the same conclusion, and that includes Gerhard Heinrich and Joel Habener when they reported the sequence of the rat GLP-1. Even as late as 1986, Mojsov, Holst, and their collaborators reported that GLP-1 is a peptide of 36 amino acids. But when I reviewed the sequence of the pre-pro-glucagon in my laboratory at the endocrine unit at the MGH, I noticed immediately the presence of this arginine residue at position 6. And this was shown much better on the next slide. This is the arginine residue. And if this arginine amino acid is cleaved, then the biologically active sequence of GLP-1 will start at position 7. You can see how I was counting the amino acids which will lead to the active sequence. And in the next slide, I will show you in more detail my rationale. So this is 1 to 37, and we compare it to the sequence of glucagon. We can see that there is very little homology between the amino acids in the two peptides. But if arginine is being cleaved by enzymes, then we'll have 7 to 37. And if we compare it to the glucagon sequence, we can see, as shown here in the vertical lines, a perfect alignment between the amino acids in 7 to 37 with those in glucagon. I knew from my previous work at the Rockefeller University the importance of the histidine amino acid at the beginning of the sequence. If that was removed, then glucagon became inactive. And I hypothesized that 7 to 37 would be the biologically active sequence, and it's going to be the incretin. Now, what are the incretins? They are secreted by the intestine, they stimulate insulin release by the pancreas, and the insulin release is glucose dependent. So the first question which I asked was, well, is 7 to 37 secreted in the intestines? And in the next slide, I will show you the general strategy which I developed to prove that 7 to 37 is incretin. So the first step was to develop, to obtain by the solid phase synthesis large amount of 7 to 37, and to use that synthetic material in all the subsequent studies, which included to develop methods to detect 7 to 37 in intestines, and to use and to characterize its biological effects. For its biological effects, we used the perfused rat pancreas to measure whether 7 to 37 stimulates insulin release. I also collaborated with Dr. Joel Habener's group and his postdoctoral fellows, Dr. Dan Drucker and Jacques Philippe, to study GLP-1 expression and biological effects in cell lines. But those experiments were not conclusive, and I will not show them. And then finally, we used the synthetic 7 to 37 in clinical studies. So let's start. So the first step was to obtain synthetically 7 to 37 and 1 to 37. I used both peptides in all the experiments. And to do that, I used the manual shaker, which was invented by Professor Bruce Merrifield. In 1983, manual shakers were not available commercially, and Dr. Merrifield sent me one of the shakers from his laboratory. So the next step was to use the synthetic peptide 7 to 37 to develop very sensitive radioimmunoassay, which is a quantitative measurement of the concentration of 7 to 37 in tissues and in blood. The next step was to develop sensitive chromatographic methods to separate 1 to 37 from 7 to 37 and from other peptides which are present in the intestine. And for that, I use a gel filtration chromatography, which separates on the basis of molecular weight. And as you can see here on the right panels, I did detect GLP-1 in the rat intestinal extracts. But gel filtration chromatography was not sensitive enough to separate the two molecular forms of GLP-1 because they have very similar molecular weight. But they differ by charge. As I showed you before, there is the 1 to 37, there is an arginine amino acids, and there are there is aspartic acids and 2 glutamic acids. And I used at that time the very newly developed high-pressure liquid chromatography, which can separate by charge. And as you can see on the right panel, I separated 2 of them, and most importantly, I actually found 7 to 37 in the intestines, and that proved the first point. 7 to 37 is secreted in the intestine. Now to answer the second question was then, does 7 to 37 stimulate insulin release? And for those studies, I started a collaboration with Dr. Gordon Weir at the Joslin Diabetes Center in Boston. And when we infused very small amounts, 10 to the minus 11 molar, we found very sharp increase in insulin. Now, 10 to the minus 11, it's a very, very low concentration. It's the same one which is found in the intestine and in the blood. And this was much more pronounced as a 100-fold higher concentration. But we see here that as the concentration of 7 to 37 increases, so does the fold stimulation of insulin. It's almost 20-fold. Now, we also used the 1 to 37, and we found that it's not insulotropic, doesn't stimulate insulin release. Even at 10 to the minus 9 concentration, we see here, this is now concentration of 1 to 37, and even at much higher concentrations. So we proved the second point, that 7 to 37 stimulates insulin release from the pancreas. And in subsequent experiments with Gordon Weir, we showed that its stimulation of insulin release is glucose dependent. As you can see here, as the glucose concentration increases from 6.6 molar to 16.7 molar, we have increase in insulin release. Note here the difference in the units on the y-axis. And this is exactly what would you expect physiologically, that insulin release will increase when after a meal or if glucose is taken orally. Another important conclusion from this experiment was that 7 to 37 is not active at low concentration of glucose. And that indicated that any medicine which will be developed on the basis of 7 to 37 will have beneficial effects on suppressing hypoglycemia, which is typically observed with insulin administration. So then, we proved that 7 to 37 is an incretin. It's secreted in the intestine, it's released by the pancreas, and the release is glucose dependent. And of equal importance is that it proved that its glucagon gene encodes the incretin. Now, other investigators in the field were interested in the same problem. And at the beginning of 1987, Holst, Orskov, and their collaborators reported that another form of GLP-1, 7 to 36 amide, stimulates insulin release at 10 to the minus 9 molar fourfold. Notice the units on this, on the y-axis. With Gordon-Weir, we detected that glucagon also stimulates insulin release at the same 10 to the minus 9 molar concentration, and it is fourfold. And in contrast, 7 to 37 was much more potent. It was a 20-fold stimulation in insulin secretion. And that, of course, remained even at a very low picomolar concentration, as I already showed you. So I was interested in understanding more the biology of 7 to 36 amide. The difference between the two peptides is only in one amino acid. It's the arginine at position 36, which is being amidated. And using the same analytical methods which I showed you before, we found that 7 to 36 amide is secreted in the rat intestinal tissues. And then, in collaboration with Gordon-Weir, we showed that 7 to 37 and 7 to 36 amide are equally potent in stimulating insulin release at these very low concentrations. Gerhard Heinrich, working independently in Habener's lab, showed that identical pre-pro-glucagon messenger RNAs are shown in the pancreas and in the colon. But the northern blot analysis is not sensitive enough to show which peptide are released in these tissues. One needed the analytical methods which I developed to show that in the intestine we have a secretion of different forms of GLP-1, the two active forms, 7 to 37 and 7 to 36 amide, and the second glucagon-like peptide 2. While in the pancreas, glucagon is being secreted, and to a much smaller degree, the GLP-1 peptides. Now, clinical studies followed very quickly, and by the end of 1987, Kramer and his colleagues in Stephen Bloom's lab reported that 7 to 36 amide is incretin in men. We were interested in understanding whether 7 to 37 will stimulate insulin release in patients with type 2 diabetes. And in collaboration with Dr. David Nathan from the Massachusetts General Hospital, we showed that administration of 7 to 37 leads to a very high increase in serum insulin and lowering of plasma glucose levels. And this experiment showed for the first time that the therapeutic potential of 7 to 37 to treat the disorders of type 2 diabetes. Now, our results were very quickly confirmed, first by the group of Suad Efendic and his Scandinavian collaborators, and then a year later, but by Michael Nock in Creutzfeldt's lab. Now, I was interested in understanding whether 7 to 37 has biological effects beyond pancreas. And for those studies, I looked at the distribution of the GLP-1 receptors in human tissues. And with my colleague, Yang Wei, we found that identical GLP-1 receptors are expressed in the pancreas, in the brain, in the heart, and the kidney. And our results suggested that if there is a medicine which is based on the active sequence of 7 to 37, then it can be used to treat multiple disorders in human health. But 7 to 37 had a very short half-life in circulation. It was very quickly degraded by an enzyme dipeptyl peptidase 4. It removed the first two amino acids to lead to come with a compound or analog which was inactive. Now, several pharmaceutical companies develop small molecule inhibitors of the enzyme. The first on the market was sitagliptin, and it's an oral medicine, and its main advantage is that it prevents the hypoglycemia, which is typically observed in with treatment of insulin. Now, the scientists at Novo Nordisk increased the half-life of 7 to 37 from 2 minutes to 13 hours to 1 week by incorporating chains of lipids to the active sequence of 7 to 37. And this is what now being used this is what's now known as liraglutide or semaglutide, and both compounds are being used for treatment of diabetes and obesity. The next slide shows the number of 7 to 37 analogs, which are now used for treatment of diabetes and obesity. The ones in blue are the ones which I showed you for earlier developed by Novo Nordisk. And the newest newcomer is tirzepatide, which is distributed by Eli Lilly, and it's based on the work of doctors Richard DiMarchi and Matthias Jopp. So in conclusion, GLP-1 analogs represent the first example of when a single medicine can be used to treat multiple human disorders, from diabetes to inhibition of appetite, weight loss, and possibly even Parkinson's disease. We also know that it has protective cardiovascular effects and also beneficial effects on the kidney. So I expect in the future we will see a development of other peptide-based therapeutics which can be used as treatment for multiple human disorders. So this is my acknowledgement slide. First of all, I want to thank the Rockefeller University for all their support. I want to acknowledge my mentors, the late Dr. Bruce Merrifield and late Ralph Steinman. I also want to thank the Howard Hughes Medical Institute and the late Dr. George Cahill for their support of my initial work on GLP-1. Now, I'm very indebted to my collaborators for the studies on understanding biology of glucagon and GLP-1, and I didn't have time to show you those. At the Massachusetts General Hospital, I want to thank my colleagues, Dr. Henry Koitman and Dr. Henry Cronenberg, who was the head of the endocrine unit when I did my studies with GLP-1. I also want to thank the group of my very talented and dedicated research assistants who work with me side by side on the bench. And of course, Gordon Weir's collaboration was critical for understanding the biology of GLP-1, and I want to thank him and the members of his group for those studies. And finally, below are the funding agencies which supported my work on GLP-1. Thank you very, very much.
Thank you very much to Professor Mojsov. And finally, we would like to invite to the stage Professor Holst to deliver his lecture. Ladies and gentlemen, please join me in giving a warm welcome to Professor Holst.
So now you will see a live lecture. Thank you very much for inviting me to give this talk. And I'm sorry for all of you to be forced to hear the same story a third time, but I'm going to give it a slightly different angle, as you will see. Let's move ahead. This didn't do anything. This did not move it. So you have to do something about this. Otherwise, it will be very boring. Yeah, but it doesn't do it. Okay, so the problem I was facing, and we are now back in 1972 when I was a young surgeon, working with patients that had been operated for gastric ulcer disease, was reactive hypoglycemia. And here's an example of these patients that you can see have developed a very low glucose after the operation during a meal. So this is reactive hypoglycemia. And it's clearly due to insulin secretion, as you can see here. And so the question was, what causes this hypersecretion of insulin in these patients? And of course, the incretin effect that you've already heard a lot about could be a possible mechanism, and it could be one of the incretin hormones. The problem was that the incretin hormones were not known in those days. So we knew that it was an endocrine mechanism, as shown here. This was the first study of the incretin because of the recent insulin radioimmunoassay that had become possible. But which hormone could do it? So we looked at these cells in the gut, and these are endocrine cells in the human gut, and almost all of that I will show you will be in humans and a little bit in pigs also. So these cells are found in the human body, and they react with antibodies against glucagon. And that's, of course, ridiculous because glucagon is supposed to be in the pancreas and not in the gut. So glucagon was interesting if it's really in the gut, because glucagon in these patients with reactive hypoglycemia, if you inject it, would stimulate insulin secretion to a large degree. So could it be they released glucagon and could therefore explain the reactive hypoglycemia? So it turned out, when we looked a little bit closer, that there were two sorts of glucagon antisera. So some of them would react with extracts from the gut, as indicated before, and you can see here's a radioimmunoassay curve where the extract from the gut reacts very strongly in the assay, but some of them were very specific and would not react. So there was something different between the two sorts of glucagon, that in the pancreas and that in the gut. And if you use these antibodies for radioimmunoassays against in human meals or glucose tolerance tests, as you can see here, those reacting with the gut extracts would show a big increase, and those that only reacted with the pancreatic glucagon would show a decrease. So there was obviously a big difference here. So we hypothesized, and this was in 1973, that this material from the gut glucagon, was actually responsible for the reactive hypoglycemia. And you can see here, we have the insulin responses in these patients, the hypoglycemia, and also the secretion of this gut material. So we thought that this was a good idea. This might be the responsible factor. We started to look for it, and it took about 10 years to find out what it was. And eventually we found out that this was glycentrin. Glycentrin is shown here, and in the middle it has the entire glucagon sequence. As you can see here, it's a peptide of 69 amino acids. And we looked at it and said to ourselves, okay, this is really interesting, because these bits and pieces that are outside glucagon, we could also find them in the pancreas. And we could also, using a tool I had developed, the perfused porcine pancreas, we could also show that they were released from the pancreas. So exactly the same structures were released both from the pancreas and from the gut. So we said, okay, glycentrin is likely to be the precursor of glucagon, the pro-glucagon molecule. It's just processed differentially so that glycentrin is produced in the gut and glucagon in the pancreas, plus the little bits that are sticking out here. That was published in Nature, and that's one of my very few Nature papers in 1981. Okay, so the next slide shows that we also looked at pro-glucagon, because we also felt that it was hugely interesting to look at this because of the possibility of other products. And it turned out that pro-glucagon had to be bigger than glycentrin. And cell-free studies suggested that it might have a molecular size of up to 18,000, which is much more than glycentrin with its 69 amino acids. And in Don Steiner's laboratory, they had isolated a fragment here that was 10,000, which did not contain glucagon at all. So this had to be that part of the non-glucagon part of proglucagon. So what was in the rest of proglucagon? That was the interesting question. And here, Graham Bell in 1983 cloned the hamster proglucagon sequence and afterwards the glucagon gene from people. And here's the cDNA, the predicted, the cDNA structure. And this shows that, yes, glycentrin was exactly here, and that is exactly the structure of glycentrin, and with glucagon in the middle. But in addition, his studies showed that there was a glucagon-like stretch of the cDNA here and a glucagon stretch of the cDNA here. And that was, of course, extremely interesting for us. So we immediately had those stretches synthesized from the predicted structure and also created radioimmunoassays for them, like here, like this. Okay, and then we started extracting a lot of peptides from tissues, and this is what the result of all that study you see here. And what we found very, very clearly was that there were extractable peptides in the gut and in the pancreas with GLP-1 radioimmunoreactivity, but they were processed differently. So you have already the glycentrin in the gut here, but in the pancreas you have glucagon, right? But this stretch with the GLP-1 and the GLP-2 in the pancreas is collected in a single molecule. We call it the major proglucagon fragment, and we could demonstrate by chromatography that this was collected in one single peptide, whereas from the gut, the two sequences were separate, as you can see here, GLP-1 and GLP-2, and we did gel filtrations to demonstrate this, and Svetlana Mojsov is correct. We could not separate a 1 to 37 from a 7 to 37 with that method. But we nevertheless could demonstrate not only that they were formed differentially in the two tissues, but we could also demonstrate that they were secreted because we were using isolated perfused pancreases and isolated perfused intestines. And when we stimulated the intestines, we could demonstrate the release of GLP-1 and GLP-2 separately from the gut. And we presented that in Madrid in 1985 for the first time. And here I'm showing you one of the materials that we are looking at. That is my favorite tool. That's the isolated perfused pig pancreas. And it's doing in this slide exactly what a pancreas is supposed to do. It's producing pancreatic juice, of course. But it's also producing hormones, as you know. So we could look at the effects of these peptides on the pancreas. And we did that. And we took those synthetic peptides and put them on the pancreas. And you can see there was no effect of either GLP-1 or GLP-2 at all. So that was, of course, extremely disappointing. So instead, we said, let's try and see what we can find in the extracts. So we made extracts of human and porcine gut using our assays and a lot of chromatography and all that stuff that you used in those days for isolating peptides, and then had them purified and sequenced, and now we could put them on the pancreas to see what they would do, and we could also make a sequence analysis of them. So that's what we did, and this was presented for the first time in April 86. So here we have the result, and you can see here that this natural material, this natural extracted material, this is natural GLP-1, very powerfully stimulated insulin secretion from our perfused pancreas. So this was really the day of the discovery. And here, and we just published that, we presented it at the EASD in Rome in 1986, and you know, you had to send it in before April to have them accepted, so this was what happened. So eventually we were able to map precisely all the products of proglucagon in people, in man, and as you've already seen, the peptide that was found in the natural form was the truncated peptide GLP-1 78 to 107 amide. And now you've been confused about this amidation business, but it turns out that in people, almost all the GLP-1, more than 90%, is amidated. It doesn't matter terribly much, but it's very important for the measurement of GLP-1 because most best assays use this amidated form for measurement, so it's pretty important anyway. And so the real structure of human GLP-1 was presented for the first time in 1989, where we were able to use mass spectrometry and more advanced methods to demonstrate exactly the structure. Here you see, again, insulin secretion from the isolated perfused pancreas with GLP-1. And you can see here at different glucose levels that at low glucose levels, it doesn't do anything. At normal glucose levels, stimulation. And then the increasing responses in a very nice dose-response relationship here with increasing glucose concentrations. But that was not all, because there were many peptides that could stimulate insulin secretion these days. So it was not really that exciting. And anyway, you know, the incretin effect didn't work in people with diabetes, and GIP was inactive and all that. So it was not that exciting, but this was exciting. This was inhibition of glucagon secretion. So this is, again, the isolated perfused pancreas. And here we are giving it GLP-1, and you can see the very powerful inhibition of glucagon secretion. So this was interesting because, you know, regulating glucose, you have insulin to downregulate when glucose increases. You have insulin secreted and will downregulate the glucose by these functions. But if glucose starts to fall, then you have glucagon instead here. That will increase the glucose. So by attacking both insulin secretion and glucagon secretion, you will have a double effective mechanism for regulation of blood glucose. And this is exactly what you see in people if you infuse them with small amounts of physiological amounts of GLP-1. You can see here that glucagon is decreasing, that insulin is increasing, and as a result of that, you have a decrease in the hepatic glucose production because of these two changes. And because of that, you will see plasma glucose starting to fall. But it doesn't fall very much. It only falls about 1 millimoles. Then the glucose dependency sets in, and there's no more any effect on insulin secretion and no more any inhibition of glucagon secretion. So it's self-limiting. So this is exactly what happens in people. The story about the reactive hypoglycemia, let me complete that, because it turns out that the exendin 9-39 is a very effective blocker of the GLP-1 receptor. And if you block the GLP-1 receptor and then look at these people with the reactive hypoglycemia, you can see that you can completely prevent it, and you can also prevent the insulin secretion. So that story was correct. It was GLP-1 that was responsible for the reactive hypoglycemia of our patients. Okay, so we were able to identify GLP-1 as an incretin hormone. Could it have additional physiological? You know, you're standing there with a white powder in your hand and you're asking yourself, what does it do? So insulin secretion was, of course, one thing. So in the following decade, we mapped all the actions of the new hormone in people. So here you have the first effect that we were able to see. That is the inhibition of gastric acid secretion. This is the emptying of the stomach. Emptying of the stomach. And you can see with GLP-1, there's a strong delay of gastric emptying in people. And here you're looking at acid secretion. And again, you can see that it will inhibit acid secretion very strongly, particularly together with PYY, but that's a different story. Here's the control experiment. So it's a very potent inhibitor of acid secretion. And that was really interesting in these days because you were interested in acid secretion. And interestingly, it is particularly effective on vaguely stimulated, neurally stimulated acid secretion. And that means that the effect is exerted via the brain, via the central nervous system. And that's really interesting. So here is another important effect that we found. That is inhibition of food intake. So here you see the inhibition of appetite between two meals. So satiety is increased, hunger is decreased, and as a result, you eat less at a subsequent ad libitum meal. So this was the first demonstration of the inhibition of food intake in people. And here you have a dose-response relationship from a meta-analysis that we did a couple of years later. And you can see that the more you infuse people with GLP-1, the less they will eat. So there's a clear dose-response relationship here, which is important, as we will see later. So is it a physiological action? We can again look at the effects of the GLP-1 receptor blocker. And you can see here, if you block the GLP-1 receptor, you will eat more, which indicates that you have been inhibiting, that the hormone has been inhibiting food intake in the control experiment. So how does it do that? Is it a direct effect on the stomach because of the inhibition? No, it's not. Could it be a central effect on the brain? Yes, we think it is. And even more importantly, it could be interaction with sensory afferent neurons. And so how does it look? This is a GLP-1 cell in people again. And you can see that it sits here in the bottom of a crypt. And out here you have a red structure, and that's a capillary. And that capillary is stained with this enzyme, which is called dipeptidyl peptidase 4, that cleaves GLP-1 molecule. And you see here what's happening. So these two amino acids are cleaved off, and this remaining part is completely inactive. And we found that this happens with an incredibly fast half-life. So you have a half-life of GLP-1 in the body by 1 to 2 minutes. So what really happens is this. You have the GLP-1 here, the cell. GLP-1 is secreted and crosses across the lamina propria here to the capillary, meets the enzyme, and is destroyed. Only about a quarter of what is released makes it to the liver. In the liver, there's again DPP-4 that will cleave half of what arrives at the liver. So now you're down to 12% of what was released. And there's also a circulating DPP-4 that will degrade some of that. So very, very little reaches the pancreas in the intact form. But what can happen instead is that GLP-1 may interact with vagal sensory afferents here, they do express the GLP-1 receptor, sends signals to the brains, to the nucleus of the solitary tract up here, and either signal further on to the motor nuclei of the vagus or directly up to the hypothalamus, where we have the appetite controlling centers. So this is what may happen. Can we demonstrate that this happens in people also? That's, of course, a little bit difficult, but we had these tools and we had these patients with truncal vagotomies, so we could look at them and see what would happen if we were cutting the vagus nerves. So we did that. So here is an experiment where we're looking at gastric emptying in these patients, and in healthy controls, you can see this is the normal emptying curve, and this is with GLP-1, and again, you see the very powerful inhibition of gastric emptying with GLP-1. In those that had vagus cut, oops, I'm sorry. This is what I should do, yes. In those that had the vagi cut here, there's absolutely no effect on gastric emptying. But more importantly, if we look at something that is really dependent on the vagus, that's the pancreatic polypeptide response. Every pancreatic polypeptide cell seems to receive a cholinergic fiber from the vagus. So you can see the response here to the meal, here, before, and after GLP-1 completely obliterates the response. In people with vagotomies, there's absolutely no effect of GLP-1. And then comes the food intake experiment, of course, where we in the controls can see the inhibition of food intake, whereas in those with vagotomy, there was no effect. So again, this supports very strongly that GLP-1 also in people acts via the vagus nerve. So there's another entry into the brain in people, and that is via the circumventricular organs. And here we demonstrated already in 1996 that if you inject GLP-1, this is in rats, but it was radioactive so that we could do autoradiography labeling and look for where does it go? Where in the body does GLP-1 go? And we found it in two places here, in the subfornical organ or in the area postrema in the brain. So it can reach these circumventricular organs of the brain. That's a very important pathway into the brain. And if you look at C-FOS, that is activations of the neurons, you can see that the neurons in the area postrema are activated by injections of GLP-1. This was liraglutide, and there's also some activation in the nucleus of the solitary tract. So there is a receptor there, there's binding there, there's activation of the neurons. And this is true for several regions. Amygdala is one of them, and some of the hypothalamic nuclei also show activation after this interaction in the area postrema and the subfornical organ, and also in the median eminence. So there's a difference between what the exogenous GLP-1 does and the endogenous GLP-1. So the endogenous normally will act via the vagus nerve and hit the brain. If there's a very strong stimulation so that the circulating concentrations are very high, remember that it's destroyed by DPP-4, so the concentrations are very, very low. There is the effects via the vagus nerves, but also some effects via the circulation. But the injectables, the agonists here, they will mainly work via the circulation and then reach the receptors in the brain. So putting everything together, we have a peptide here that will regulate and improve all aspects of beta cell function. It may even impair, or it may even act on the reduced beta cell mass. It will regulate glucagon hypersecretion. It will regulate overeating and obesity. We'll come back to the complications, and it will also counteract insulin resistance. So it was really, really interesting to see what might happen. And here we have the experiment. This is the experiment. This is when Michael Nauk was able to, together with us, to infuse GLP-1 intravenously for 4 hours in patients with rather severe diabetes. They came to hospital with a glucose concentration of 13 millimolar. And in the course of those 4 hours of infusion, you can see that glucose concentrations fell to completely normal levels. This was really a major discovery. And this was because of stimulated insulin secretion, as you can see, and inhibited glucagon secretion, and they returned nicely as glucose concentrations were normalized. So this was really the start of the diabetes therapy with GLP-1. And we later proved that this was actually possible as a chronic therapy also by infusing people for 6 weeks with GLP-1 using insulin pumps. And this is what Medisandra here did. And you can see that we had fabulous effects on glucose. A1C was reduced by 1.3%. We had weight loss. We had improved sensitivity and enhanced beta cell secretion and no side effects in that study. So that really proved that GLP-1 was a possible therapy for type 2 diabetes. So we had this problem that it was degraded by the DPP-4 enzyme, of course, and I told you that it had a half-life of 1 to 2 minutes. And not only that, but the kidneys will also extract GLP-1. So for that reason alone, it would have a half-life of 5 minutes. So this is what happens if you inject people subcutaneously with GLP-1 at the highest tolerable dose. You can see that very, very little survives in the active form, and most of it is degraded. Just a few % survive. So this was, of course, interesting. So we suggested instead that after having demonstrated that this was the way it was destroyed, that perhaps inhibitors of DPP-4 could be useful for the therapy of diabetes. And this is one of our greatest successes, I think. So here we demonstrated in pigs that we could infuse them with GLP-1, and this is the intact form, this is the metabolite, and you can see that it's hugely degraded in spite of the intravenous infusion. Then we inhibit the enzyme, and now you can see all is protected, and what happens? What happens is that we threefold increased insulin secretion in this way. So this demonstrated that you could really protect the endogenous GLP-1 with DPP-4 inhibitors. And as a result, in people here, if we did this and infused a DPP-4 inhibitor, this was possible very early on, you can see that we could increase the concentrations of active GLP-1, surviving active GLP-1 by 2 to 3-fold. Actually, GIP was also protected, and glucagon was inhibited very nicely. So this was picked up by the pharmaceutical industry very quickly, and these long-acting DPP-4 inhibitors, vildagliptin, sitagliptin, and saxagliptin, were developed that had a 24-hour duration, which turned out to be essential for this effect. And this has become, you can see here, the first clinical study by Warren, beautiful study, showing that in the controls, there was an increase in A1C over a year, but with the DPP-4 inhibitor, there was a decrease to down around 7% over that year. So it is really a very nice and effective therapy. And you know, the DPP-4 inhibitors have been sold all over the world since then. It's really a huge success. So again, this problem with the degradation, because the other way to solve this problem was, of course, to find stable molecules and use them instead. And that's what was done. And here we have the series of available stable compounds. These two are short-acting ones, and for various reasons, they are not very suitable for modern therapy at all. But here we have the long-acting ones. We have liraglutide, which is acylated, has a fatty acid chain. It binds to albumin and survives in that way. And here we have semaglutide with another fatty acid. I was there to help them develop this molecule. It was a great success. It was really, really exciting to build this weekly molecule. And here you have dulaglutide from Lilly. And then you have tirzepatide that you've already heard about, which is a mixture of GIP and GLP-1, which is really, really interesting. And I'll show you some examples of that here. So these are studies in people with type 2 diabetes with tirzepatide, which is a very, very active, let me call it a GLP-1 agonist, because that's what it is. You can see what happens here. So 50%, more than 50% of those with type 2 diabetes that are treated with this dose of tirzepatide will develop an A1C level below 5.7%. 5.7% in A1C, that is normal. That is normal. So that's really a sensational result. As dramatic and as sensational was this finding with semaglutide here. This is from a cardiovascular outcome trial, and you're here looking at cardiovascular risk. And this is the LEADER trial with liraglutide, actually. 10,000 people looked at for up to 5 years. And you can see that their risk of developing major adverse cardiovascular events is reduced by 13%. And if you look at the meta-analysis of the compounds that are now available, you will see that apart from the short-acting ones over here that do not work, all of them show this improved action on MACE, major adverse cardiovascular events, that is stroke, myocardial infarction, and cardiac death. And if you look at the individual components of that, MACE, here, myocardial infarction, stroke, cardiovascular mortality, all-cause mortality, and even heart failure are all improved. The risk is reduced for all of them. Here is stroke. This is a composite study with all the semaglutide studies that were done here, and you can see that stroke risk is greatly reduced. And this is perhaps one of the most important things because stroke is really difficult to prevent and treat. So how is that possible? And you've already heard some about all these mechanisms, but let me point out one thing first of all. That is the effect on inflammation, because it really does reduce the levels of inflammatory markers and inflammation in general during therapy. And let me also emphasize the effects on the vasculature, on the capillaries here, where we have receptors for GLP-1. And I emphasize these two mechanisms because they are general for the whole body, of course. So that could explain why there are multi-organ effects of the GLP-1 agonists on so many organs like the brain, the heart, the kidneys, and the liver. So that is perhaps the most important reason for that. Come on. Now it has really stopped working again. Here. So let's look at the body weight now. So how did it become a weight-losing agent? We had already shown, as I demonstrated for you, the dose-response curves for GLP-1 on food intake. But how did it become a body weight? So it turns out that liraglutide was effective with a small weight loss of a couple of kilograms, which was nice, of course, but not tremendous and not surprising. But what happened then was that visionary people said, okay, let's try to do a dose-response study and see if we increase the doses of liraglutide further, that we can obtain bigger weight losses. And here is what you get. So now in this study, where you go up to 3 milligrams of liraglutide, normally you add in 1.8 or 1.2, you can have weight losses of up to 10 kilograms. So that paid off, and that was the foundation for registration studies, the so-called scale studies for the development of liraglutide for body weight, the Saxenda compound. So now comes the other one, the semaglutide, which is improved in many ways, and the improvement resulted in a duration of action with a half-life of 1 week. And it was improved also on insulin secretion and on food intake. So it's really, we were very proud when we sent that molecule off. And again, it caused a weight loss, which was nice, of course, but again, could it be improved further? And that was the question. So let's see. Here's a dose-response study again. Again, courageous people said, let's try and see if we can increase the dose and get even bigger weight losses. So here is the placebo. Here is the liraglutide, yeah. And so comes the different doses of semaglutide here on top. And now we're getting at a weight loss of 14% of body weight. So this formed the foundation for registration studies for semaglutide. And we have the STEP trials. And I'll show you one example here. It's a STEP trial, step 4. And what we see here is a weight loss of 18% because of this. So again, tirzepatide is also very effective. And here you see with tirzepatide a weight loss of up to 26%. So now we're approaching the weight losses of bariatric surgery. And we already saw the results of this SELECT trial, where 17,600 people were looked at for up to 4 years, 40 months, yes, and where there was a 20% reduction in cardiovascular events during this study. So my overall conclusion is, GLP-1 is an important incretin hormone. It has effects on food intake, on appetite. It acts via afferent neurons and the CNS. GLP-1 agonists are effective anti-diabetic agents. It has protective effects on the cardiovascular system and offers renal and neuroprotection. And I believe that the GLP-1s will change obesity and diabetes therapy from now on. Thank you.
Thank you very much to Professor Holst. And now let's invite Professor Wang back to the stage to give us the conclusion. Welcome.
Thank you for the three laureates to give us a wonderful talk. As you can see, that a discovery can come from several angles. And three laureates, they approach the problems from, let's say, in Joe Habener's case, studying using a biochemical and molecular biology and cell biology approach. And later on, of course, Mojsov used this chemical approach to prove that the 7 to 37 peptide is important. And then finally, I think Dr. Holst here gave us a wonderful talk on, from a point of view of a really clinical aspect angles, and to not only prove, but later on, he contributed in making the semaglutide a long-lasting GLP a drug here. And so this is really a wonderful achievement now. I think everybody in the audience can remember this word GLP-1. It's a very important drug, not only in the weight loss, in anti-diabetics, and now many more. Even, I haven't heard cancers, but we do, huh? Oh, okay. So in cardiovascular diseases, dementias, and so on. So it becomes a magical kind of a drug. Now, this is an interesting issue here. Now, you know, in Taiwan, I heard that even though it's a prescribed drug for diabetics, but I understand that in some cosmetic clinics, they sell it to what we call the rich ladies. And it's called a skinny needles. So it turns out that it has been all brought up by those cosmetic use, and in the hospitals, we are shortage of those drugs for diabetic use. So this is a kind of phenomenon as unexpected, but I think now with this Tang Prize, the promotion of the GLP importance, I hope you will not go further in that direction. But anyway, so this is truly a wonderful event here, and I'm sure maybe GLP-1 will go beyond Tang Prize or Lasker Award and we'll go somewhere else. So we will look forward to that. And I think I want to take this opportunity to thank all the participants here and go and promote GLP-1. And, and we, the lectures here will not have this kind of a Q&A session, but I hope if you have any particular problem or questions, you can ask Dr. Holst here, and later on we will have a press conference, and then you may be interested in joining that. Thank you very much for joining us.
Thank you very much to Professor Wang for the conclusion. Thank you. Ladies and gentlemen, this concludes the Laureate Lecture for Biopharmaceutical Science. Actually, Professor Holst has to leave now for the press conference. So, ladies and gentlemen, please join me in giving another warm round of applause to Professor Holst. Thank you so much for giving us this wonderful lecture today.